to next speaker, and next speaker is Filo Sanchez Rodriguez. As part of joint project between University of Liverpool and Diamond Life Source, he currently engaged in PhD under supervision of Professor Daniel Regen and Gwan Yard Evans. His work focused on development of new computational tools for determination of protein structure, with particular interest in the application of residue contact prediction and machine learning to molecular crystallography. Over to you.
Thanks. Can you hear me?
Yeah.
Yeah, cool. All right. Yeah, so thanks for that nice introduction. And yeah, I would like to start by thanking the organizers for giving me a chance to talk in front of you about the stuff that we've been doing recently at the University of Liverpool, particularly about how we can use evolutionary covariance-based predictions for model validation. So I thought it would be a good idea to start with a brief introduction about what residue covariance is and how it can be used to predict residues that will be in contact with each other. So say you're looking into a protein of interest and there's a residue that mutates. Now, if, for example, because that residue is nearby a catalytic site of the protein, it's possible that that mutation may alter the functional shape of the protein. When this occurs, it's possible in some cases to observe that residues that are neighboring that residue that is mutated may also mutate in such way that will counterbalance the original mutation and the overall functional shape of the protein will be retained. Now, when you put a lot of distant homologs into a multiple sequence alignment, it's possible sometimes to observe these coordinated changes between residues that are neighboring each other in the folded protein. And this is what we call residue covariance. So what would you do? What you would do then, you would put this sequence alignment through powerful statistical methods, which mostly these days would be machine learning, and you would be able to predict how likely it is that each residue pair is in contact with each other. Once you get those predictions, usually the way people represent these things is in the form of contact maps, which is usually a representation where you have in the vertical axis and in the horizontal axis the residue number, and then you just put a dot if you think those residues are in contact with each other. Something some of you may have heard about are histograms. This is the latest thing in the field, and it's something that, for example, the latest ab initio modeling pipelines, like for example AlphaFold, make use of, and it's a new type of prediction. So the input will be the same. It's still a multiple sequence alignment of distant homologs. But in this case, we go one step further. And what happens is that we no longer predict whether the residues are in contact with each other. What we actually predict is the actual distance between those residues. The representation of this kind of predictions is called histogram, as I said. It's very similar. So again, on the vertical axis and in the horizontal axis, you could get the residue number. And then in each spot, well, each spot is colored correspondingly with the distance predicted by the residues. So the brighter the spot is, the closer those residues have been predicted. And it's not apparent at first, but the truth is these predictions actually contain a lot of information that are very useful in different fields of structural biology. So for example, if you are trying to characterize your target, you could use them for domain boundary predictions. If you're into molecular replacement, you could use them as well for search model selection. The idea here would be that a good search model should, in theory, satisfy a lot of the contacts that have been predicted for your target. Something that you could do as well is sequence tracing. So say, for example, you have your model, you're trying to trace the sequence, but you have a part of the model where the resolution is low, so it's difficult to trace the sequence. You can help yourself with contact maps just to be able to sequence, so to trace the sequence. And now for the main topic of the talk is how can we actually use these predictions for model validation? So what we do here, what we're doing all the time is comparing predictions with observations and trying to find an agreement or disagreement between them both. So let's start with this simple case. It's just a helical pair. So on the right, we'll have our observations, which is just what we start with. So we start with an electron density map, and then you will trace your model, which in this case, as I said, is just a helical pair. Then on the left, we have the predictions, which basically it all starts with the sequence of your model. Then you will put that sequence through a pipeline, like for example, TR Rosetta or AlphaFold, and you will get your predicted contact map. So a prediction of what residues are in contact with each other. Then assuming your predictions are correct and your model is correct, what you will expect to see is an agreement between them both. So a lot of contacts that are being observed in the model should be in the prediction and vice versa. However, let's look into these different cases. What happens if, for example, there's a mistake in the model? So here we introduce a kink in one of the helices. then again, assuming your predictions are correct, what you would see is a lot of contacts in the helical pair that are not present in the predictions, and a lot of predicted contacts that are not present in the model, which is shown here in red. So this is the concept behind our pipeline, but we can detect things that are not just modeling errors. We can also detect more, well, smaller things as well, such as, for example, register errors. Let's take a look into this other case. This is an artificial case we've been working with just for development purposes. It's easy to work with. This is just the region of interest of that model. It's a bigger model, but I'm just showing this part. In gray, I've colored the model, and then every other residue is colored with distinctive colors so that it's easy to see what's going on with the sequence register. So what we did, we made one of the loops a little bit shorter, as you see here, and just to push the sequence a little bit farther and to induce that sequence register error. And you can see, for example, this blue serine is now somewhere here, and just push the entire sequence of that central beta strand. And again, if we just go back to the idea that I just explained about comparing predictions with observations, you can imagine that if we were to compare the contacts on the right, so on the model with the contacts on the left, so the prediction, you will see a lot of mismatches, which is exactly what happens. So we take the predicted contact map, the observed contact map, and we superpose one on top of each other. This is the representation we'll get. So in black, we can see the contacts that match. In red, the ones that are present in the model, but not in the prediction. And in gray, the ones that are present in the prediction, but not the model. Now, in terms of metrics that we have actually developed, what we would do now is go in a residue by residue basis. We calculate things like false positive rates, false negative rate, accuracy, etc. And what all of this means is just counting how many contacts we see in the model that have not been predicted, how many contacts have been predicted that are not present in the model, this kind of thing. And you can imagine that, for example, in this region, which is actually the region where the error is, these residues will have a very high false positive rate, a very high false negative rate, low accuracy, low sensitivity, and this is all because there's actually a problem going on in the model. Other metrics that we have been looking into as well actually relate to distance predictions. So this is the thing I was mentioning at the very beginning. These are predictions between residues that actually involve the distance. So here we have the histogram of the full model. And again, we do exactly the same thing. We take the predicted distances and the observed distances in the model. We compare them, and then we'll get this heat map, where the brighter the red is, the higher the difference between the predicted distances and the observed distances in the model. And again, you can clearly see that there's something going on around residue 80, which actually coincides with the place where the sequence register error has been induced. In terms of metrics, here what we'll be talking about will be the RMSD, which is just an RMSD between the observed distances in the model and the predicted distances, weighted by the confidence of each prediction. So just to summarize, as I said, we compare the model with the predictions, and we extract at each residue number in the model, we extract a series of features. So we will get their RMSD, false negative rate, etc. So what do we do then? Well, we take these features and we put them through a train SVM classifier that at each residue position will predict a score of how likely it is that that residue is part of a model error. The higher it is, then the more likely it is that that residue is in a model error. Just as an interesting note, actually the way we trained this classifier was using errors that we found in the EM modeling challenges that took place between 2016 and 2021. The reason why we used this is just because we wanted clean data, so a curated database where we could actually find errors that we were sure that these were errors, so that the classifier was trained with clean data. In terms of performance, the classifier achieved an accuracy, sensitivity, and a specificity above 80% after training, so I think it's rather good. And also, something we've been looking into as well is actually how can we fix those errors once we detect them. And some of you may have noticed in this case that you have a lot of red contacts here that are observed in the model but have not been predicted, and a lot of gray contacts here that have been predicted but have not been observed in the model. And if you pushed the sequence here just by about four residues, these contacts in red would match perfectly the contacts in gray. So we will be able to fix that error. And this is exactly what we do in our pipeline as well. So we take both contact maps and we try to align them, trying to find the contact maximum overlap between them both. And once we find the sequence register that gives you the perfect overlap between predictions and observations, we compare that register with the register of your model. And then if we see there's a difference there, we could say, well, all right, there's an error there. And if you want to fix it so that the predictions match your model, you should assign this sequence to your model in this particular region. So again, just a summary, in our pipeline, we'll have two different sources of information. On the first hand, we have this classifier that at each residue position, it will give us a score, just so that we are able to detect the error. And once we detect the error, then we align the contact maps, trying to find the CMO, so contact maximum overlap register. And if we see that it's different to that register of the model, then we just provide with a fix for your errors. We're able to detect and to fix the error. So where is this implemented? Where is it available if you want to use? Well, first of all, it's available in Concrete, which is our Python package for covariance information. It's got a command line tool. It's very easy to use. So you just type Concrete validate, then you give your sequence, your predictions, and your model. And then it will produce this figure that I'm showing here, where in the vertical axis, you have the smooth score. This is the score produced by the classifier. And then in the horizontal axis, you will have the residue number. Then we set up a threshold at 0.5. So whatever it's above 0.5 has been predicted as a model error. And it also has these two color bars at the bottom. So the first one, which is blue and red, is similar information. It's again whether the classifier predicted that particular residue as being part of a model error or not. And the bottom bar, it's blue or orange, and it corresponds with the contact maximum overlap I was talking about. So that's the contact map alignment. And it's blue if there's a match, so the sequence register is okay. It's orange if it's not okay, so there's a mismatch, and we provide with a fix for it. On top of that, all this information is printed in the terminal. So you would get a table where each residue is telling you this is the score, or this is the probability that this residue is in a model error. And this is how you can fix it if we have a fix for it. Also, something we have been working on, it's implementing this into Iris in collaboration with John Akure at the University of York. Some of you may know Iris. It's this new graphical user interface I've seen before. It's very nice, it's very easy to use, and it will produce these concentric circles where each circle depicts a different metric for model validation. So it looks really nice. What we've been thinking about is adding our metrics to Iris so that we can also use them there. So first we have the CMO. This is the contact maximum overlap I was talking about. And again, in green, if there's a match, in red, if there's a mismatch in the contact map alignment. And we have the covariance score, which in this case refers to the score provided by the classifier of how likely it is that there's an error. And again, you can see it there. This is a great way of using these new metrics. Now for an interesting part, this actually relates to the previous talk that Greg made. And some of you may remember that he was talking about how he used his pipeline checkmass sequence to look for register errors in about 6,000 structures deposited in the PDB using cryo-EM at a resolution of 5 angstroms or better. And as he was saying, the thing is we were developing these methods completely different concepts that are completely independent from each other while looking at the same problem, but in a completely different way. So we thought it would be great if we could also take a look in those structures and see if we can find the same errors that he found in a way that we will be able to validate each other and just to provide, basically it's just a way to be sure that those errors are actually there. So as he was saying, we were able to validate more than 300 errors across 6,000 different structures in the PDB, which is what I'm showing here. This is a scatter plot where each point corresponds with a register error that we found. The size and the color of each point corresponds with the residue shift that goes all the way from 1 till 5 or higher. Vertical axis will be the number of residues that were affected in that register error. And in the horizontal axis, you will get the resolution of that structure. So a few things that are interesting. First of all, we were able to find a lot of errors in the PDB, but I think you can start to see some interesting patterns. For example, register errors with a smaller shift, like for example, only one residue also tend to affect the smaller portions of the structure, so with 50 residues or less. And you can almost draw a line about 100 residues, whatever is above that, it tends to be bigger errors as well, so with a bigger shift. But I think here really the takeaway message is that we have these new, two new tools that are great. They complement each other and they work really well. And I think really now the message is trying to convince the users to use this tool to look at their structures, try to validate them, and try to look for these errors that are not deposited in the PDB. And just a few conclusions now. So yeah, so what we've been doing from the very beginning, so what we're doing in our pipeline all the time is just trying to compare predictions with observations, just trying to find agreements or disagreements between the predicted distances and the observed distances in your model, using this classifier that was trained with the EM modeling challenge, with the structures deposited in the EM modeling challenge. And then we do the sequence reassignment, right? So we try to align the predicted contact map and the observed contact map, trying to look for the contact maximum overlap sequence register so that we can give it to you and you can fix your error. So we detect the error and we fix it. And then if you would like to use this, it's available in Concrete and then also in Iris. And just to finish, I would like to thank my supervisors at the University of Liverpool, Dan Ricklen and Ronan Keegan, my supervisors at Diamond, Rinda and Melanie, all the people at Liverpool, Adam, Javier, David and Shah, Greg for the data set, IEMBL Hamburg, and then at the University of York, John Aguirre for the implementation in house. Thanks a lot.
Thank you. Thank you for your talk. And we have a question in Slack. Does one need many bacterial autologous sequence, or does this work with some dozens on eukaryotic autologous?
So in theory, I mean, there's nothing like, it does not depend. So you could use it with any sequence that you would like to use it. The thing here is that I guess he refers to the multiple sequence alignment to obtain the covariance predictions. So here, the thing is that basically the more homologs you're able to accumulate into the multiple sequence alignment, then the more precise your predictions would be. So that's what we call the multiple sequence alignment depth. So it would just be the number of effective sequences in your multiple sequence alignments. So the more that you can accumulate, then the better they will be. But yeah, that's it really.
And also, can it be used to validate quantitative contacts, like protein-protein contacts between two different peptides?
Sorry, can you repeat that?
Yes. Can it be used to validate quantitative contacts, like protein-protein contacts between two different polypeptides?
So yeah, so at the moment, you can only do it per chain. So you can only validate one chain at a time. You wouldn't be able to validate well, I mean, the thing is, what we do is we validate the chain, right? So what you would need is intra-chain contact predictions to validate your chain.
And I think, we have first hand, Emmett. You want to ask a question?
Yes, I have a question. Let me ask it. So my question is, can this method apply to multiple models more than two? So if you have, like, for example, 10 models and you want to find the errors in this model and also the agreements between all of these models to combine them in a one model?
It could be used. It's not something we provide, but say, for example, you have these models and you want to validate all of them, then obviously you could take ConKit, for example. As I said, it's a Python package, so you could even write a small script and you can run this on all the 10 models and try to see where the errors are. But it's not something we provide, but you could do this.
Okay, thank you.
And the last question, are the distance in the diagram sensitive enough to distinguish between types of contact? Like hydrogen bonds are defined by donor-acceptor pairs, but results also. Yeah.
So in this case, when we talk about covariance between residues, what we're really talking about is just residue-residue contacts, right? So when we say that two residues are in contact with each other, what we're really saying is that the carbon betas are within 8 angstroms of each other. So that's all that I mean with contacts, really.
Thank you.